Thank you, Sam. All right. I presented when I was a fellow and it freaked me out so badly. I died, but I came back for more. So congratulations on getting this far. It's been a tough competition. 75 abstracts came in and we had to be really mean and say, if you didn't come in at the right time, we kicked you out. So you've done very well to be selected. We have, how much do we have? 19,000 in prizes, including travel grants for the winner, not including travel grants, and 2 PhD top-ups. So hang around at the end for the prize giving. So as you all know, and I've been screaming at you over the morning, get your friends, register and vote for you for People's Choice. Okay, so we also like to thank in the afternoon, we have guest speakers, Keith, who's a mental health nurse from, is it from Cairo? He started off Keith's Closet, which is a resource for clothes for patients on this area that's been admitted for mental health problems. And Dr. Suzanne Law, who's one of our new associate professors of nursing, she'll be giving out the cash. All right, so without further ado, let's get on with it. I'll pass you over to Jane Butler, who's the chair of the Basic Science Division. Thanks, Jane. And she'll introduce you all.
Yeah, hi everyone. I'm Jane Butler from Neuroscience Research Australia down the road. I guess we could start just first with an acknowledgement of country. We'd like to acknowledge the traditional custodians of the land, the Gadigal and Bidjigal people, and we pay respects to their elders, past, present and emerging. So this is the Basic Science Division and we have 4 speakers and everyone gets 10 minutes to talk, 5 minutes of questions. There's judges in the audience marking the talks. And yeah, the first talk we'll start with is Dr. Holly Holliday from the Brain Tumour Group in the Children's Cancer Institute. And she's talking on the targeting the epigenome through combined FACT and BET inhibition in diffuse midline glioma. Thank you, Holly.
Thank you so much for this opportunity to present some of our work into targeting the epigenome in diffuse midline glioma. So diffuse midline glioma is a really aggressive form of childhood brain cancer. As the name suggests, it grows diffusely in midline brain structures, with the most common site being the pons. This cancer is most common in children below the age of 10 years old. And unfortunately for these children, the majority of these kids will die within 1 year of being diagnosed. And due to the really challenging location of the cells growing in these really critical brain structures, surgery is not possible and chemotherapy is ineffective. The current standard of care is radiotherapy, but this only alleviates the symptoms temporarily and the cancer always comes back. And so far, there are no targeted therapies for DMG. But hopefully this will soon change because there's been some really important landmark discoveries made in DMG research, and this has led to identification of this driver histone mutation, H3K27M, which causes epigenetic disruption to the cells. So just some definitions to get us all on the same page. Epigenetics refers to, or it literally translates to above genetics. So it refers to that layer of regulation on top of the DNA code itself. So DNA is really tightly wrapped around these histone proteins, and together the DNA and the histone proteins make up the chromatin of the cell. And the packaging of chromatin allows the condensation of this incredibly long molecule of DNA to fit inside the size of a nucleus. And importantly, it allows for the correct genes to be switched on and switched off. So chromatin can exist in this open chromatin conformation, which is more accessible for transcription of the genes, or in this more closed, silenced, transcriptionally silenced state. And what happens in DMG cells, which have this driver histone mutation, is that you essentially get suppression of, or you lose suppression of this repressive histone mark, which ultimately leads to this open chromatin conformation. And this leads to the wrong genes being expressed and is a really key driver in the aggressiveness of this disease. So therefore, targeting the epigenome and transcription is emerging as a really key therapeutic strategy against DMG. And today I'll be talking about two such targets. The first one is the FACT histone chaperone. So this is a heterodimer made up of two proteins, SPT16 and SSRP1. And this chaperone is really important for maintaining the integrity of chromatin. It's targeted by the drug CBL137, which our lab has shown to have preclinical activity against DMG, which has resulted in a phase 1/2 clinical trial, which includes DMG patients. BRD4 is the other target, so it's a BET protein. It binds to active acetylated chromatin and promotes transcription of oncogenes. It's targeted by BET inhibitor drugs such as JQ1, and this class of drugs has also been shown to have preclinical activity against DMG, and these drugs are also in clinical development. So if we look at expression of FACT and BRD4 in normal and DMG cells, we can see that both FACT and BRD4 are overexpressed in DMG cells compared to normal cells, which led us to hypothesize that DMG cells are dependent on these proteins and will therefore be sensitive to combination therapy with CBL and JQ1. This study therefore aims to determine both the efficacy and mechanism of targeting these proteins in DMG. So first of all, I wanted to know, where do FACT and BRD4 bind to in the DMG genome? So to do this, I'm using a technique called cut and run to map the protein DNA interactions. And this is done in patient-derived DMG cells. So very briefly, this uses antibodies which are tethered to a nuclease, and these nucleases then recruited to the protein of interest, where it then cleaves the DNA. We elute the DNA and sequence it and visualize the binding as these sequencing peaks. So using this technique, I was able to show that FACT and BRD4 co-localize at accessible genes in DMG. So this is just a snapshot of one gene, which is the famous MYC oncogene and is a known target of both FACT and BRD4. And you can see that this FACT subunit as well as BRD4 are located at these regions of open chromatin, which is measured using a different technique called attack sequencing. So this is just one gene, but this is generalized across the whole genome. So now we're looking at all transcription start sites, and the red color indicates more binding. So these transcription start sites have been ranked by how accessible they are based on the attack sequencing. And what I want you to see is that the FACT and BRD4 are enriched at these open accessible transcription start sites. So this implies that they're important for maybe promoting or maintaining open chromatin and transcription. So we wanted to know what happens when we target them. More specifically, I wanted to know what happens to the structure of chromatin and the expression of genes. So to do this, I've treated 2 patient-derived DMG cells in 4 treatment groups, untreated, each drug alone and the combination, and performed attack sequencing to measure open chromatin and RNA sequencing to measure gene expression. So again, this heat map is looking at all transcription start sites in the genome. This time, the more red in color indicates the more open the chromatin. And what you can really appreciate is that in the combination treatment cell, we're getting a decrease in chromatin accessibility, which seems to be driven mostly by the FACT inhibitor CBL137. So again, taking the MYC gene as an example, you can really see that the combination treatment is decreasing accessibility or closing up the chromatin at this gene. And when we do differential accessibility analysis, we found that the majority of regions were actually condensed rather than open. And importantly, when we look at the genes encoded by these regions, in this heat map here, the majority of these genes were actually switched off. So I wanted to delve further into what kinds of genes were being regulated by these drugs. So now I'm talking about the RNA sequencing results, and these volcano plots here show the significantly differentially expressed genes, with the downregulated in blue and the upregulated in red. And both drugs alone led to thousands of genes becoming differentially expressed, but this was the most exaggerated in the combination treatment. So next I wanted to know, what are these genes involved in? So to do this, I've performed some gene set enrichment analysis, and this was done on genes which were common to both of the cell lines. So starting off with the downregulated genes, these were involved in DNA replication and repair, RNA modification, histone modification, and ribosome biogenesis. So these pathways are really important for the growth and division of cells, and these are being switched off by drug treatment. On the other hand, the upregulated genes were more involved in stress and transport, which could reflect a more acute response to these drugs. The cells responding to the drugs. So finally, I wanted to integrate the 3 different data sets. So I took the genes which were bound by FACT and BRD4 in the cut and run, had closed chromatin when they were treated with the inhibitors, and decrease in expression. And this generated a list of 14 genes, which we consider high confidence targets of these factors. And I'm in the process of validating these, but I just wanted to highlight one of these genes, which is KLHD8CA, which was very recently this year been discovered to be an epigenetic driven oncogene in the context of adult glioma, where it drives a stem cell phenotype. So do these changes in chromatin and transcription actually do anything to the cells? So the answer is yes. So this is looking at a combination dose response curve, and you can see that the combination of the 2 drugs was more effective than each drug alone. And this is in the context of cells which have the histone mutation. But when we delete the histone mutation, this effect is actually lost. So this is telling us that there's something about the chromatin conformation in these histone mutant cells that is sensitizing the cells to the drug treatment. And what's really exciting is that this drug combination also works in animal models of disease, of DMG. So this is work led by my amazing colleague, Amina Khan, and she was able to show that the combination treatment significantly prolonged the survival of mice in 3 different DMG models. So to conclude what I've presented today, I've shown you that FACT and BRD4 bind to regions of open chromatin. When we inhibit them, we get the closing up of chromatin around those transcription start sites and deregulation of transcription, including downregulation of some of these really interesting target genes. This leads to a synergistic effect both in cell lines and in animal models of disease, where we're getting an extension of survival. So therefore, FACT and BET inhibition is a promising new combination treatment for DMG. And with that, I'd like to acknowledge everyone in our lab, particularly my supervisors, David and Maria, and Amina Khan for leading the animals aspect of this work, as well as everyone else in the group, the bioinformatic team for their support, and our external collaborators and funding bodies. Thank you.
Okay, that was great. Thank you. Perfect timing as well. Are there any questions from the audience? You've got to talk in the microphone so the Zoom people.
Hi, Holly. Nice talk.
Thanks.
Wonderful work. JQ1 is a pretty old BET inhibitor. Is there any plans to use a more clinically relevant BET inhibitor, and are they also able to cross the blood-brain barrier? And I guess that's one of the key aspects of any treatment for DIPG, and just your thoughts on what the next steps are regarding this combination.
Yeah, you're right. So we're using JQ1. It's like a classical BET inhibitor. We're using it as a tool compound for the mechanistic studies. We are also looking into clinically relevant BET inhibitors. One of them is CC90010, which is in clinical trial for brain tumors. So it can cross the blood-brain barrier. So we're also looking at this drug in combination with CBL in the animal studies as well. Yeah.
Okay, so I'll just keep asking questions because clearly no one else has any interest in this, but I do. So the closing of the chromatin, I mean, what's driving that? I mean, the H3K27 mutation is still there, right? So the PRC2 complex can't act on that particular histone mark. So, you know, you show they're closed. Do you have any thoughts on what would be driving that repression, or is there, you know, like, what's happening? I mean, you showed no binding, but is it real? And I guess the ATAC showed it's not as open anymore, but what do you think's happening there?
So I have looked at histone marks, at least on the global level, just looking at the methylation and acetylation. At the same time point that I did these ATAC experiments, not seeing any changes globally. So what I think might be happening is that the FACT complex is somehow maintaining this open chromatin. And then when we come in with the CBL and inhibit it, it's just causing like condensation of the chromatin. And I think in combination with the BET inhibitor, which is driving transcription, so it's like this combination of the closing up of the chromatin driven by CBL and the suppression of transcription driven by JQ1, which is causing the effects. But to pinpoint the actual mechanism with the histone mutation is proving a lot more difficult than I thought. So I've also done cut and run for the histone mutation, and it does co-localize with FACT and BRD4 at some sites, but it's definitely not a generalized thing across the whole genome. So yeah, still really trying to pinpoint the interaction between FACT and the histone mutation. And it's sort of like, so originally I thought that FACT was chaperoning the histone mutation and it was a more direct effect, but now I think that the histone mutation is causing this chromatin conformation, which is sensitizing or is making the cells dependent on FACT and BRD4. It might not be a direct inhibition of the histone mutation, but it's just creating a chromatin conformation that makes them more susceptible to drug inhibition.
Lovely. Thanks, Holly.
Thank you.
Thanks, Holly. That was lovely. And for a person that doesn't understand a lot of cancer research, that was great to be able to understand it. But there's lots of people that you're acknowledging there. So what was your role specifically in this project?
Yep. So my role was mostly the mechanistic molecular side of things. So the cut and run technique I introduced in our lab. So it's an alternative technique to a technique called chip sequencing, which I tried but didn't have much luck. So I had to abandon that technique and try the cut and run technique. So that's been sort of one of the main roles that I've had, as well as the attack sequencing and RNA sequencing and the bioinformatic analysis. Not all of it, like I can't do the alignment of the reads, but I'm doing more the downstream biological questioning of the data. Yeah.
Okay, thank you very much, Holly. Give her a round of applause. And we'll move on to the next speaker. So the next speaker is Isobel Williams from the School of Psychology at UNSW, and she is speaking about reversing the effects of prenatal opioid exposure, the efficacy of sodium butyrate in an animal model. All right, thank you.
Thank you. So in the past 20 or so years, there's been a rapid increase in opioid use in pregnant women. And this is particularly concerning for their fetuses who are exposed to opioids across that period of prenatal development. In the short term, we expect they're more likely to be born preterm, but they're also likely to experience the symptoms of opioid withdrawal at birth. So currently, when symptoms are severe enough, we can treat these symptoms of withdrawal by offering these children additional opioids. However, in the long term, what does this exposure across prenatal development and potentially across that early postnatal period mean? So in terms of the physiological outcomes for these children, we know that they seem to have poor long-term outcomes in terms of their brain development. We see that they have decreased brain volume and particularly decreases in communication in the white matter tracts, suggesting an impairment in myelination. And then in terms of their peripheral development, these children have changes in their immune functioning, so they have an increase in inflammatory markers, and they also have changes in their gut microbiome composition and gut functioning. And there's a wealth of literature that would suggest that changes in immune functioning and gut microbiome composition during this sensitive period of development has long-term impacts on myelination, behavior, and cognition. And indeed, we see this come to play out with these children who have poor long-term cognitive outcomes. So they have a 50% chance of diagnosis with a mental health condition by the age of 5, and they have poor performance at school. So even when we look at them compared to socioeconomically relevant controls, we see that they're consistently falling behind. And this pattern of falling behind continues into adulthood, where these children have an increased risk of drug use and an increased risk of suicide. So it's really a problem that's persisting across the lifespan. So what we know so far is that opioid use during pregnancy is an increasing problem, and these children go on to have impaired brain development, particularly in terms of myelination. They have altered immune system functioning, and they have changes in their gut health and microbiome composition. So, and they have poor long-term outcomes in terms of behavior and cognition. But what can we as preclinical researchers do? So luckily, we have this really excellent rodent model of prenatal opioid exposure. So this model really nicely recapitulates a lot of the changes that we see in the infants. So we see these rats who've been exposed to opioids before birth have poor brain development, particularly in myelination. They have changed immune functioning, and they have changes in their gut health. And they also show these long-term changes in behavior and cognition. So we can use this model to unpack these deficits and these changes that are happening, but we can also use it to explore potential treatments for this growing population. So when we were looking for treatments, the one that we came across and chose to explore was sodium butyrate. So butyrate is a postbiotic. It's produced by the breakdown of fiber in the gut, and it's known to have these pro-myelinating, anti-inflammatory properties and to improve gut health. And importantly, it's also a widely available dietary supplement, so you could buy it from your local chemist. So exploring this sodium butyrate as a potential treatment is exactly what we did. We got our sodium butyrate and we got our pregnant rats, dams, and we exposed them to sodium butyrate in their drinking water and the opioid methadone via a mini osmotic pump consistently across gestation and lactation. And this is a period that mirrors prenatal neural development in human infants. And the offspring of these dams who've been exposed across gestation and lactation were then tested on a range of physiological and behavioral tests in adolescence and into adulthood. So just quickly go through some of our early data because I don't have time to go into it in depth. We saw that the dams, their weight didn't change depending on the treatment. And if they had sodium butyrate, they drank a little more. So they seemed to quite like it. The pups at birth, they had reduced birth weight, but their weight gain was accelerated across adolescence or infancy if they'd had opioid exposure, which mirrors what we see in the human population. And we also saw that in this early developmental period, methadone-exposed pups had increases in anxiety-like behavior, kind of mirroring again what we see in opioid-exposed children. However, we didn't really see many impacts of the sodium butyrate in this early developmental period. But when we moved on and kind of looked at a bit later, so the beginning of adolescence, and we looked at neural development, what happened was when we looked at our marker of myelination in the hippocampus, we found that methadone had reduced the amount of myelination present, and this was reversed when they've been offered additional sodium butyrate treatment. Then we looked at our astrocytes as a marker of kind of neuronal health, blood-brain barrier functioning, and again, we found that there was a reduction in the number of astrocytes in the hippocampus that was reversed when additional sodium butyrate had been given to the dams. And then when we looked at microglia as our primary immune cell of the central nervous system, we found that administering sodium butyrate increased the number of microglia that were present in the hippocampus, indicating a potential change in immune functioning in the central nervous system. So the other system that I've been talking about a lot, the gut microbiome, we also looked at in the dams and in the pups, and this work was done by our collaborators, Josie and Keith. And what we found was in the dams, being exposed to methadone resulted in a decrease in the alpha diversity of the gut, so kind of a shift towards a more unhealthy composition, with a trend towards improved composition in rats that have been additionally exposed to sodium butyrate. And we also found that the methadone-exposed ones had a decrease in firmicutes that are required for the production of our treatment, butyrate. And then in the pups, we looked at their microbiome at the beginning of adolescence and then also in adulthood. And we found that methadone exposure resulted in an increase in bacteria associated with diet-induced obesity, inflammation. And then we also found that sodium butyrate exposure resulted in a decrease in bacteria associated with inflammatory conditions, suggesting that methadone is resulting in changes in the microbiome composition associated with decreased gut health, while sodium butyrate seems to be resulting in changes associated with improved gut health and decreased inflammation. And then we looked at these rat siblings in tests of behavioral tests in adulthood. So these tests took place over 40 days after any drug exposure had ended. So they weren't being exposed to the sodium butyrate or the opioids. And we used this 5-choice serial reaction time task to measure attention and impulsivity. So when we looked at our indicator of impulsivity in the rats, we found that if they've been exposed to the opioid methadone, they made more impulsive responses, but this was reversed if they've been exposed to sodium butyrate, suggesting that methadone is increasing motor impulsivity, but this is being reversed by additional exposure to sodium butyrate. And when we looked at our measures of sustained attention, we found that methadone-exposed animals made more incorrect responses and they missed more trials, suggesting impairments in sustained attention that was again reversed in both cases when they'd had additional treatment with sodium butyrate, suggesting that the sodium butyrate reversed this impact. And then in these rat siblings, we also looked at a task that's assessing working and spatial memory, the trial unique non-matching to location task. And we found that if the rats had been exposed to methadone, they took longer to learn the task. But again, this was reversed when they had additional treatment with sodium butyrate. And then when we increased the working and spatial difficulty of the task, we found that the methadone exposed rats made more incorrect responses, which again was reversed with additional exposure to sodium butyrate, suggesting that methadone is impairing working and spatial memory, but this is being reversed by the sodium butyrate exposure. And then kind of mirroring what we saw in the previous task, we saw that rats that have been exposed to methadone again made more impulsive responses, but this pattern was again reversed by additional treatment with sodium butyrate. So this work provides a link between prenatal methadone exposure in an animal model, changes in gut microbiome composition, long-term changes in neuronal health and development, potential changes in central nervous system functioning, such as our microglia here, and long-term changes in behavior and cognition. We are hypothesizing that this could be due to increases in gut permeability that allow more toxins to enter the bloodstream, which are more able to cross the blood-brain barrier following opioid exposure. And I've started kind of pursuing this pathway, looking at the pups' blood-brain barrier permeability shortly after birth, and it seems that methadone may indeed be increasing that barrier permeability. So whilst it's unlikely that there's any one mechanism that underlies all the deficits associated with prenatal opioid exposure, this research provides some evidence that sodium butyrate, a widely available dietary supplement, has the potential to improve the children around the world who are being exposed to opioids before birth. And I'd just like to thank my supervisors and all our collaborators. Thank you.
Thanks so much, Isobel. That was really interesting. Any questions from the audience? Okay, I'll start with Julie.
2 questions. Thank you, Izzy. How would you translate that onto a human? When would be the best time to give sodium butyrate?
Yeah, absolutely. So it's interesting that a lot of the changes that we saw were later in development. So we're actually now pursuing that. Could it just be that postnatal treatment is going to be effective in reversing the deficits, especially if we're thinking about a clinical population? When are they going to be accessing care? When is our treatment going to be effective? And I have some really early data that suggests that offering the sodium butyrate after the opioid exposure has ended is also having positive long-term impacts on cognition. Yeah, so we're seeing decreases in impulsivity that are really similar to what we saw with the prenatal exposure. So it seems like quite a wide window for potential treatment. Yeah.
Yeah, it's very intriguing. Like this one agent can have so many like pleiotropic effects.
Yeah.
But I just noticed, so is it 3% weight per volume that you're giving these rats for ad libitum in the water?
Yeah, it's in the water.
So my question, I guess, reflecting Julie's, is the translatability. Like, how much sodium butyrate do you have to give? Like, I don't know what 3% weight per volume equates to in a human who might weigh 55 kilos, etc. But then, and then on top of that, it seemed to me like in the memory test, in the sham, there was, for the incorrect responses, sodium butyrate almost seemed to trended like they did poorly. So what's that window where it helps?
Why is it helping?
And then maybe potentially making it worse for people who don't have it. So the translatability in that therapeutic window, have you got any thoughts on that?
So when we went with the 3%, that's really the highest concentration that's been reported to be tolerated in rats. So as our first pass, we were kind of like, let's just give them as much as we can and see what happens there. We've now, in our next study, also decreased the dose because I ran a dosage study where I found that if they actually had half the concentration, so 1.5%, they drank double the amount. So they end up having the same total exposure. So they seem to like it more. But because of the way rats to humans works, you have to give humans considerably less of a drug than you do to rats because of the speed of their metabolism. So the amount of opioids that we're giving to the rats is also kind of roughly 10 times the amount that humans. So how much? Yeah, so I haven't reverse engineered it, but it's a reasonable amount. So when I ran the dosage study, I was making sure that the dose fell within the recommended dietary intake. It's not an insane amount. Yeah, I was making sure that it aligned with recommended amounts of usage in human patients, because there's some evidence people are exploring the use of sodium butyrate during pregnancy for diabetes, and so kind of was following that literature and checking that we were following a similar level of exposure. And then in terms of why it seems to be having negative impacts in the sham animals, it's a really interesting question that we're kind of trying to unpack. It often seems that offering a treatment during this sensitive period when it's not needed has negative impacts. So I was at a conference recently and people offering other probiotics, other kind of chemical treatments, they see these negative impacts in the sham animals. So it seems like you need this insult for it to balance out, but I'm not entirely sure why that is. It could also be rat specific, that they found it stressful, that you wouldn't necessarily have that problem with the humans. Yeah.
Is there time for one more question? So it's lovely presentation, fantastic, thank you. And we saw all these positive impacts on the rats in the dams. Were there any null results or negative results that you couldn't present?
Sure. So I think in terms of the immuno specifically, the changes seem to be quite specific to the hippocampus. So I looked at some kind of lateral ventricle area, and there really wasn't the same level of obvious change. So it seems like the hippocampus might be a bit of a hot spot, which is interesting, especially given the tasks that we use really are spatial memory dependent. So that's aligning nicely. The next step will be to look at the prefrontal cortex and kind of see if that taps into our impulsivity attention tasks. But I think really I've been very, very lucky. Like things seem to have worked out reasonably well. We did look at peripheral inflammation at a later time point, so after exposure had ended, and we found that there really weren't many changes in peripheral inflammation, but we're hypothesizing that that change is happening earlier, and then it's kind of calmed down by the time we were looking at it, but we're seeing the shadow of that in the neural development. Yeah. You can always talk to me.
Do you want to
be around?
quickly
Yeah.
ask, wanted you to have a chance to say your role in the project?
Oh, my role in the project. Sure. So I helped design the project from the kind of outset with my supervisor, Kelly, and then I collected and ran all the animals and did all the immuno, ran all the behavioral tests, analyzed all the data, apart from the microbiome, which was done by Keith and Josie, very kindly, our collaborators. Yeah.
Okay, thank you so much.
Thank
everybody.
you.
Thanks, Isobel. Okay, so our next speaker, we have a change in the order of the program. The next speaker is Binod Rayamajhee, and he is speaking on the role, this one's a harder one for me to say, the endosymbiotic Pseudomonas aeruginosa in the development of Acanthamoeba keratitis in an animal model. Okay, thank you.
Thank you. Good morning, everyone. I'm very excited to present a part of my PhD on topic endosymbiotic Pseudomonas aeruginosa, how it impacts on the development of Acanthamoeba keratitis. Just imagine how you feel when there is no eyesight. So I'll be talking about how these devastating eye infections cause the ocular infections. Let me start with a brief introduction of the Acanthamoeba. It's an opportunistic free-living amoeba which is present in different ecological habitats, including our domestic tap water. And it can invade our body through different routes, for instance, eyes, ulcerated skins, and nasal passages. The unique feature of the Acanthamoeba is it adapts biophysical lifestyles, including the trophozoites, which is metabolically active and large. It contains the Acanthamoeba, which helps to move the organism from one place to another place. And it includes another dormant phase, that's the cyst. It has two wall, ecto and endo walls, and it can remain viable for many years, even in harsh conditions. That makes the infections relapse and severe as well. Acanthamoeba keratitis is an important infection of oculi, and it primarily affects the contact lens, where because contact lens, it behaves as a mechanical vector to transmit the Acanthamoeba from environment to our eyes. Due to non-specific clinical features of Acanthamoeba keratitis, it is always difficult to diagnose, and very limited drugs are available to kill both the trophozoites and the cyst. That's why nearly 30% of the patients, they require surgical intervention to control the infections. And sometimes the infection, it becomes very severe, and it lasts for months to years, and they are required to remove their whole eyes. These are some photographs shared by the Acanthamoeba keratitis patient on different social media, just to raise the aware about the Acanthamoeba keratitis, as it should be heard by the broader scientific community. Another unique feature of Acanthamoeba is this is an environmental phagocytic predator. It engulfs or predates a wide range of the microbes, including some of the potential human pathogens. This is an example of the Acanthamoeba bacteria association, how it behaves in the environment. Basically, the uptake of the intracellular bacteria, it starts with the receptors that present on the cell membrane of the Acanthamoeba, and it starts with the phagocytosis and the formation of phagolysosomes. Basically, most of the bacteria, they are digested within the phagolysosomes, but some of the bacteria, they can prevent the phagosome-lysosome formations, and they can survive intracellularly in the Acanthamoeba. And interestingly, some of the bacteria, they are expelled into the external environment in the form of the expelled food packets in the number of hundreds to thousands. If they come in contact with the in our eyes, at that time, they can cause the infection with the host Acanthamoeba. These days, more severe form of Acanthamoeba keratitis with longer treatment duration and poor treatment outcomes are being reported globally. And this may be due to the infection with Acanthamoeba that containing the intracellular bacteria. So the overarching hypothesis of my study is that the severe infection of Acanthamoeba keratitis is because of the intracellular bacteria that contain within the amoebal host. This is the overview of the methodology that we have adapted in our lab to study how the intracellular bacteria behaves in the Acanthamoeba keratitis in an animal model. This begins with the collection of the samples from the keratitis patients. They were recruited at Sydney hospitals. Meantime, we have collected the swabs from the contact lens and the contact lens solutions and from their domestic tap water as well. Followed by the pure culture of those samples and microscopy to identify the Acanthamoeba. Then we did series of the assays to identify the presence of intracellular bacteria within amoebal host, for instance, fluorescent in situ hybridizations, transmission electron microscopy, and then we did the sequencing to identify both the Acanthamoeba and the intracellular bacteria. Then we developed the animal model of Acanthamoeba keratitis using those isolates, one with intracellular bacteria and another without intracellular bacteria. We used two different isolates. Those were isolated from the Sydney hospitals patients. One was isolated from corneal samples and another one was isolated from the domestic tap water. Interestingly, both of the isolates, they were very close genetic alleles, and they belong to the subgenotype T4F, and T4 genotype is one of the common genotype that is causing the Acanthamoeba keratitis in clinical setting. Interestingly, one of the isolates that labeled as AK1 that was isolated from Acanthamoeba patient's domestic tap water that contained intracellular bacteria. As you can see here, the bacterial cells inside the amoebal host. And that bacteria was identified as Pseudomonas aeruginosa, which is a potent agent of keratitis. And that was confirmed by the multi-top mass spectrometry and sequencing of 16S RNA. The intracellular presence of the bacteria was further confirmed by transmission electron microscopy. As you can see here, those bacteria, some of the bacteria, they were undergoing the binary fissions inside the phagolysosome membrane. And the bacteria was highly motile, showing the swimming motility as compared with the wild type STEN-PO1. And that bacteria was classified as cytotoxic as that contained the type 3 secretion system gene EXOU. And we were not sure whether that bacteria was still live inside the host or not. For that, we used the Alcadella, which is a bioarchaeological probe that binds with the live bacteria inside the host. And as you can see here, that those bacteria, they have uptake the color of that bioarchaeological probe. Means for the first time, we were success to confirm those bacteria were still live inside the intracellular bacteria. And meantime, that was confirmed by our transmission electron microscopy, where we have seen the binary fission undergoing inside the Acanthamoeba host. And we developed the animal model of Acanthamoeba keratitis following the transcorneal inoculation of Acanthamoeba suspensions. For that, we inoculated 2 microliters of the suspension that contained nearly 10 to 12 of the amoebal suspensions, putting the surgical needle nearly 30 degrees to the stoma under the dissection microscopes. And then we used we divided the rats in 2 groups. Group A received Acanthamoeba only, and the group B that receives Acanthamoeba with intracellular bacteria. In their right eyes, leaving left eye as a control. And this is the overview of my experiments. After 1 week of acclimatizations in the rats in the animal facility, we started infection from day 1 to day 5, and we did a slit lamp every day to observe the keratitis progress over the course of infections. And after the day 6, we collected the eyes to do the downstream histopathology investigations. Daily infection monitoring was based on the corneal staining using the fluorescence and then corneal opacity, discharge, as well as the infiltrate. These are some representative slate images of group A rats that was infected by the Acanthamoeba only. And these are some representative cell-line images of the group B rats that was infected with Acanthamoeba with intracellular bacteria that was Pseudomonas aeruginosa. During the course of infections, as you can see here, the clinical score of the group A rat was also increased, but compared to the group B, it was obviously less. That was significantly less compared to the group B, where we have observed much more severe form of the keratitis. And then we did the histological studies, and this one is the control eye. It shows the normal and clear morphology with the intact endothelium and the epidermis, as well as the stroma in the left eyes. And in group A rats, we saw slightly disorganized epithelium and endothelium and RBCs as well as necrosis in the stroma. While in case of the group B rats, we observed the complete collapse of the epithelium as well as endothelium, and the stroma was completely filled by the RBCs necrosis. It indicates that in presence of the intracellular bacteria, Acanthamoeba can be much more virulent, and it can cause the severe inflammation as well as the tissue damage in the cornea. Then we try to re-isolate the Acanthamoeba as well as intracellular bacteria from the group B rats using the corneal homogenates. We observed the trophozoites in the corneal homogenates even after the day 6 of the infections, but the number was reduced by nearly 18 to 25 times. Meantime, we also able to re-isolate the Pseudomonas from the corneal homogenate of group B rats, while there was no growth of Acanthamoeba from the group A rats. Interestingly, in case of the trophozoites that we have recovered from the corneal homogenesis, no intracellular bacteria were observed in the fish. Means all the bacteria during the infections were expelled in the eyes, and that led to the severe infections in group B rats. And to confirm whether the severe infection was because of the combined infection of Acanthamoeba as well as intracellular bacteria, we did the second phase of the infection. For that, we used the Acanthamoeba trophozoites that were recovered from group A rats and the Pseudomonas from this one group. And these are some cell-line images of the group C rats that those were infected with Acanthamoeba alone. Those were recovered in the phase 1. And this one is, these are some cell-line images that we have got from the group D rats, and those were infected with the Pseudomonas alone. Compared to the group C, in group D, we had higher clinical meniscus, indicating that Pseudomonas is doing severe infection compared to Acanthamoeba alone, but obviously less as compared to the group B in the first phase, where we have inoculated Acanthamoeba with intracellular bacteria. And we observed, as expected, high concentrations of the neutrophil response in group B rats compared to group A, C, D, as well as control, where we saw the high concentration of the stromal neutrophils response in group B rats. This study concludes that Acanthamoeba may contain highly pathogenic endosymbiotic bacteria, like Pseudomonas aeruginosa that we have identified here. And Acanthamoeba adapted Pseudomonas showed high motility. This study shows that impact of the intracellular bacteria on the severity of corneal infection caused by Acanthamoeba as a superinfection or a polymicrobial infection that leads to extremely severe infections in animal model. Meantime, we observed that Acanthamoeba, it can release intracellular bacteria in corneal infections, which can lead to acute infections in vivo. This study offers new insights in terms of the development of more effective therapeutics against the eye-eating Acanthamoeba, with the potential to enhance the quality of the life, especially a contact lens wearer, because nearly 90% of Acanthamoeba keratitis is being reported among contact lens wearer. I would like to thank everyone who has been a part of this study, and I would like to thank Randwick Health and Innovation President for this opportunity to share my findings. I'm happy to take your questions. Thank you.
Thank you very much, Binod. And Julie.
Oh my God, that was very scary. I was Googling Acanthamoeba keratitis while you were talking. How many people do you see with this problem?
The incidence is varies country to country. In case of developed countries, it is quite high compared to low-income countries because less people are wearing contact lens. As I mentioned, contact lens is the leading risk factor. So in case of the global incidence, nearly 2% of the whole infectious keratitis includes Acanthamoeba keratitis. And in case of the Australia, nearly 30 to 40 people per year, they are getting Acanthamoeba keratitis per year.
And just the last question, what happens to them now? What is their outcome?
You mean the infection outcome?
If they have this disease?
Yes. It depends. As I mentioned, there is no licensed drug for Acanthamoeba to treat. There is no single licensed drug. So clinicians, they start with the different disinfectants, and sometimes they resolve. As I mentioned, so the symptoms, it goes or lasts for months to years. Sometimes it resolves, sometimes even it goes beyond years. So I have seen a case who was suffering since last 3 years, still not resolved. So in that case, most of them, nearly 30%, they go for the surgical transplantation of the cornea. And again, there is the relapsing cases being reported because cysts that can remain inside, in the stroma. And once they restore the new cornea, again, it will, come back. So it's really, one of the challenging corneal infection, I would say. Yeah.
I agree. I'm never touching my eyes again because I don't want to have any amoeba in my eyes. But that's the question is, and I might have missed it. So it's the combination of the Acanthamoeba plus the Pseudomonas, which is the worst, but the amoeba itself also causes damage or the disease, or is it the combination because it expels the bacteria which causes the infection, which causes the keratitis? Or like I kind of missed that logical step because you had both, right? You had the amoeba only, and then the amoeba that already had the bacteria within it.
Yes.
In your rat model.
Yes. So what we did was, because this was not done before, first time we did the Acanthamoeba with naturally occurring intracellular bacteria. What we observed was, in presence of the naturally acquired intracellular bacteria, we observed severe infection compared to Acanthamoeba alone. Itself, it can cause an infection, but in case of the presence of the intracellular bacteria, the infection has gone up.
It's gone. So yeah. If you're unlucky enough to have an Acanthamoeba which already has an internal pathogen, that's really bad. If you're unlucky to get it just by itself,
that's what we observe. Yeah.
So when you observe that, then in that case, do they pick up bacteria that just randomly goes into your eye? Is that or is it only, like, what's the process? If you get the Acanthamoeba only in your eye, in humans?
I think since our eyes, it contains some of the normal flora as well. When Acanthamoeba comes here, if it is without intracellular bacteria before in the environment, so probably it can feed in the eyes as well. If it has feeding already inside, outside in the external environment, it can go along with that one. So basically, we are saying Acanthamoeba, it can behave as microbial cargo. When it reads in the eye, we observe that it can release all the bacteria. Meantime, both endosymbiotic as well as the host, they can do the infection together, and that is really severe compared to Acanthamoeba alone and bacteria alone.
Great talk. Thank you.
Just have time for one quick question.
Yep,
Sandrino,
sure.
did you have a quick one?
So can you remind, how do people acquire Acanthamoeba?
How they do acquire?
Yeah, how does it get into people's eyes?
As I mentioned, the leading risk factor is contact lens because it behaves as a mechanical vector. When they do water activities with contact lens, at the time there is high chance of attaching Acanthamoeba. For instance, if they shower or swim with their contact lens, there is a high chance of getting attached Acanthamoeba. And in case of the low-income countries where people, they work in the farm or the fields, so ocular trauma with contaminated water or soil is being the risk factor. But in case of the developed country, nearly 90% cases are associated with contact lens. Yeah.
Okay, we have to finish up now in the interest of time, but before you go, could you just talk briefly about your role in the project, please?
So in this project, apart from recruiting patients at Sydney Hospital, most of the things I did, and for the slit lamp, I took the help from the optometrist to observe the infections, since I'm not an optometrist. Yeah.
Okay, thank you very much.
Thank you.
That was really great. Thank you, Binod. Okay, our next speaker is Nasir Shah, and I'll just. There it is. In the same vein, developing a novel in vitro flow model for endothelium using a 3D printed patient-specific arteriovenous fistulas.
Thank you very much for that introduction. I'd like to start by thanking the committee for inviting me to present at this award session. I'm really excited to tell you about one of the projects I've been working on as part of my PhD. Now, the majority of patients with end-stage kidney disease who opt to receive treatment, despite our best efforts, will end up on in-hospital dialysis. And this is a form of renal replacement therapy that requires the use of an artificial kidney machine, where we take blood out of the body, remove excess waste and water, and then pump it back into the body. And the cornerstone of this treatment is the need for reliable vascular access. And this comes in the form of an arteriovenous fistula, which is a surgical connection between an artery and a vein. Being the gold standard, we forget about the amazing physiology that we're actually creating in these patients. We take a high flow, high pressure artery and connect it to a low flow, low pressure vein. And almost immediately, both vessels experience a dramatic change in environment that's driven by this human-made shunt. Within the first 24 hours of creation, the flow goes up by 6 to 7-fold, and this continues ongoing. And to highlight this change in remodeling of the veins, here you can see 2 healthy fistulas, which allow easy access for dialysis treatments. But 50% of these fistulas will never work, and some will develop really high flows, as you can see here. And in some of our dialysis patients, this can be a big problem because cardiovascular disease is very common in our patients with chronic kidney disease, with up to 44% of patients going on to develop heart failure as part of their dialysis treatments. And in those patients that have high flow fistulas, some will shunt in excess of 50% of their cardiac output just to the fistula. So the burden of these high flow fistulas is really unknown, and the effect on cardiac outcomes is also unknown. But it highlights the importance of this vascular remodeling process and the need to balance the neo-intimal hyperplasia, which is the thickening of the vessel wall, and the outward remodeling, which is dilation of the tube. The difficulty is that the molecular basis underlying this process is unknown, which really limits our ability to not only predict, but also to direct patient outcomes. So this brings us to where my project began. I very quickly found that all the current models that are available have a lot of issues. So we started to think about novel ways to approach this. And we thought, wouldn't a unique way be to create a 3D in vitro model that uses true patient geometries that would allow us to do high throughput experiments, maintaining the really important 3D geometry of blood vessels, but allowing us to have that tight control on flow and biochemistry. And I'm terming this a macrofluidic device. The difficulty is that this doesn't exist in our area, which is why it became the focus of one of my projects. The ultimate goal is to capture patient geometries using a modified ultrasound scanner, to fabricate them using 3D printers, to then seed endothelial cells onto those models, and then to expose them to a variety of magnitudes of flow and biochemical compositions. We then hope to identify regions of interest and then to interrogate those regions of interest using pathway analysis with immunofluorescence and quantitative PCR to look at those established pathways in vessel remodeling and the inflammatory phenotype. To also use gene set analysis with RNA sequencing to identify novel signaling cascades, and then to marry all this up with the computational fluid dynamics, which the engineers are so good at doing. With any project that you're building from the ground up, there's a lot of proof of concept studies. So the first question is, can we actually modify the material we're using to grow cells? Do the cells that grow actually adhere? Are they alive? And do they respond as we would expect when they're stimulated? Here you can see modified the surface of the PDMS, which is a organosilicon elastomer. Now, this material is used in the fabrication of many microfluidic devices, but it's intensely hydrophobic, which means that cells don't like to stick to its surface. So first, you have to modify the surface before you can grow endothelial cells. Here, we tested a variety of methods of doing that, of which fibronectin coating was the best option. Next, we went on to see what conditions we needed to create a confluent layer of cells. And what you can see across this is that after modification with fibronectin, we needed to incubate the cells with 37,500 cells per centimeter squared and grow them for roughly 24 hours to get our confluent layer of cells. Here you can see that the cells that were attached were in fact alive using live dead staining. And when we stimulated these cells with TNF alpha, they increased the expression of ICAM1 and VCAM1, which is what we would expect for cells to do when they're stimulated. And what these proof of concept studies showed was that not only can we modify the surface of the elastomer to grow cells, but the cells are in fact alive and they respond as we expected. The next thing was to go on and begin fabricating our geometries. Now, as you can see here, the human arteriovenous fistulas are very complicated structures. Just to make things easy, we started with idealized vessels in the form of tubes. And this requires a little background in 3D printing. So broadly speaking, there are two different types of 3D printing technologies. There are fused deposition manufacturing 3D printers, which used a solid filament of plastic, which is heated and then extruded in a pattern to create a geometry. And there are SLA and SLS printers, which use a liquid resin, which is cured with lasers to create a geometry. For the majority of my experiments, I used a fused deposition manufacturing printer with a filament called polyvinyl alcohol, or PVA. Now, this is a water-soluble filament that is normally used as a support structure for complex 3D prints, but we actually use it as our primary printing filament to create the idealized vessel you can see on the far right. After printing the vessel, you cast it in the PDMS silicon, and then after washing it in water, as you can see, you're left with a solid block of PDMS with a lumen in the shape of the geometry that was printed. And this is the lumen that we look to grow our endothelial cells on. In parallel with this, we've also been capturing our patient geometries, and this is done using a validated technique where we have an ultrasound machine that's tagged with an optical tracking camera. And this captures thousands of two-dimensional images, which can then be uploaded into segmentation software and stitched together to create a 3D geometry. Here you can see, similar to our idealized vessels, we can print the patient geometries, initially with a block of support around them because they're very tall structures. But after removing that support, we cast them in the same way and then wash them away, leaving us with a patient-specific geometry within the block of PDMS. And all of these fabrication studies just have confirmed that not only can we fabricate the simple, but also the complex geometries, and we can do it in a way that's reproducible. As part of this process, we then went on to create our flow rig. Now, this required us to fabricate a lot of parts, and in our goal to create a sterile environment, but also to reduce our use of single-use plastics, we looked to fabricate these things using autoclavable resins. I won't go into any more detail beyond that, but I will show you on, I don't have a slide here. I will show you here that one of the pulse dampening devices that we have here is able to remove or modulate the amount of pulsatility of our peristaltic pump. So as you can see on the top, with no bubble trap, the fluid going through is very pulsy. But then when you go down and introduce the bubble trap, the fluid going through the model is continuous. This is to show that the components that we've printed are in fact autoclavable. So with a particular printing parameters, we've been able to autoclave them 5 times over and they haven't deformed from their original structure. And this allows us to create this flow rig that can be autoclaved and reused. Now, while all of these bits were being fabricated, I was initially using single-use dialysis tubing to create our first flow rig. We repurposed a gas permeable culture bag to be our reservoir of culture media. From there, the culture media flows through our models, then to a series of bubble traps, and then outside the incubator to a peristaltic pump before returning. And we were able to show that we're able to do this for about 72 hours at 200 mils a minute without the whole system exploding. This is idealized blood vessels in static conditions, and I'll just take you through each one of them. The first pane you can see on the far left is actually the PDMS silicone, which has green, yellow fluorescent beads impregnated into them, and that's simply to be able to show the borders of the actual block. The blue is DAPI, which stands for cell nuclei. The phalloidin stands for F-actin, which is components of the cytoskeleton. And this just shows that in static conditions within our 3D geometry, we've been able to grow a confluent layer of endothelial cells. This is our idealized vessels with 100 milliliters per minute of flow going over a 24-hour period. And exactly in the same way, you can see we've got a confluent layer of endothelial cells that have been maintained over 24 hours of flow. This is using a patient-specific geometry. And again, across the top, you can see a layer of endothelial cells, and going down below, it's actually a cross-section through one of the areas of the fistula itself, which is the anastomosis, just showing a layer of cells lining that geometry. I hope that I've been able to show you today that we've made a fair amount of progress towards creating this macrofluidic device for arteriovenous fistulas. We can scan patient geometries, segment them, print them, create our models, modify the surface, and grow cells on these models in both static and flow conditions. We're now at the stage where we're going to start asking this model biological questions. So the first question is obviously, does magnitude of flow affect gene signaling within these models? And also, what's the effect of a particular biochemical derangement? So in my world, uremia, which is the effects of chronic kidney disease, is that effect on flow additive, subtractive? So that's one of the first experiments we'll be doing. The second thing is also to further characterize the cells in these flow conditions, to look at polarity as well as different markers of activation and function. There's also a lot of fine-tuning of this model. We've tried to make things as simple in the first iteration, but almost every aspect of the model can be further tuned to more closely mimic true physiology. And also to further use different printing technologies. You know, even in the 1 year that I've been doing this, the type of technology available to the consumer has changed exponentially. So, you know, adapting some of these new technologies as they become available will be quite important. Finally, we know that the interaction between smooth muscle cells and endothelial cells are very important in endothelial cell signaling. So the ultimate goal is actually to go to a co-culture model. And then if we get very good at that, then maybe even doing a dual layer model. But that's, I think, going to take a little bit of time and fine tuning. I think the really exciting thing about this is that it's a model that can be adapted to any system to ask almost any question. So one of the obvious ones is obviously things like atherosclerosis and cardiovascular disease. So I think the potential is almost endless. With that, I'd like to finish up there. Thank you for your attention. I'll thank my supervisors for their guidance, the patients for their patience, and the listed grant and scholarship funding bodies for their support.
Okay, thank you very much, Nasir. We have 3 minutes for questions.
Right, I have a quick question about the elasticity of the plastic, because this is mechanico-transduction, you know, solid plastic on a dish is not physiological. So how, like I might have missed it, the actual flexibility of your actual vessel that you 3D print, how well does it model the elasticity of a blood vessel, which is obviously in skin and tissue, which is quite malleable? So
that's a great question, because fistulas are quite pulsatile. So the material we use, the PDMS, can be tuned to mimic the elasticity of vessel walls. We haven't done that in the first iteration. We've created a physical block. But theoretically speaking, you can actually mold these in such a way where you can pick the thickness of the vessel wall, and you can mix the components of the PDMS in such a way that you can make them either stiffer or more soft. So you can. We haven't gone to that stage because there's so many working parts. The idea was to make things simple first and then slowly adapt it to be closer to physiology. I mean, the jump from plates and microfluidic devices to a macrofluidic device is already a big one. So we didn't want to complicate things too much off the bat.
Thanks, that was really interesting. Can you just explain to us a little bit more about the bubble trap versus the continuous flow? Because you wanted the continuous flow, obviously, but to my mind, I'm thinking of the bubble trap and I was like, I see this pulsing, and I think, isn't that same as what you see physiologically? So why
absolutely.
do you use
So peristaltic pumps, although you can set them to be continuous, when you actually look at the flow pattern, if you set it for 100 mils a minute, it'll go between 0 and 200. And over, you know, a few cycles, it'll be averaged at 100. But that swing is very non-physiologic. So again, initially we were trying to create something that would be less complicated by making it continuous, because how do you interpret the effect of that kind of swing when it's so far from human physiology? So the idea was to try to get it as close as possible. What we're finding is that even with our best dampening devices, there still is a bit of swing. So now we're actually trying to modulate that and mimic the swings that we see in patients. So we're looking at dialysis patient fistulas, seeing where their peaks and their lows are, and calculating the difference in our percentage, and then tinkering with our bubble traps and dampening devices to try and mimic that same swing.
Okay, and could you tell us, please, your role in the study? Yeah,
so I've been actually very lucky that I was able to almost tailor this project to my interests. So I was able to capture people to let me do what I want. So from conception to actually implementation, I think along the way, every step I've been doing. The only thing that I haven't been involved in really is the computational fluid dynamics, which is almost an entire degree on its own. So the engineers do that bit. But aside from that, yeah, most aspects have been myself.
Okay, so we just should finish up there because we're out of time, but thank you again for your talk. And just before everyone goes, I've put up this last slide. There, the People's Choice Award. So you can do that with your phone and vote for the talk that you like the most. And that's a separate award to what the judges are marking. So thank you very much for coming. There's morning tea outside and we'll see you back.